I'm Gbola Amusa, and it's my pleasure to introduce our next guest from uniQure, Dr. Ricardo Dolmetsch, President of Research and Development. The format for this session is a roughly 40-minute fireside chat. If attendees have any questions during the session, feel free to ask them in the question box at the bottom, and I'll do my best to ask on your behalf. Ricardo, thank you for joining us virtually today.
Yeah, thanks so much for the invitation, and thank you all for being here. It's my pleasure. Yes.
So well, why don't you start by just giving an orientation, so to speak, to uniQure's manufacturing and CMC capabilities. Obviously, we're interested in how your approaches differ from other gene therapy companies and what advantages you might have. So feel free to take a few minutes to introduce us to the story.
Yeah, sure. So at uniQure, we have, of course, our own internal manufacturing capabilities. And we have really focused on sort of baculovirus manufacturing of AAV. So I think, are leaders in this area. We feel that there are some significant advantages to the baculovirus SF9 system as opposed to the mammalian HEC 293 system. We have developed over, more than, 2 decades, actually, this capability. And we now have a manufacturing plant in Lexington, as well as a product development group in Amsterdam that allows us to develop, a wide variety of gene therapies. We're at the moment in the process of validating our manufacturing capabilities for the launch of our hemophilia B gene therapy. We feel that our CMC capabilities have significant advantages relative to sort of the field in general. I mean, one of them for sure is that SF9 baculovirus manufacturing of AAVs is just significantly less costly than HEC 293 manufacturing, and there are a number of reasons for that. One is that the baculoviruses themselves are much less expensive to manufacture than plasmids, and so we like that. And in addition to that, of course, it is a suspension process, which means that it's easily scalable. And we can also use many of the systems that have been developed for suspension cell lines for antibody manufacturing and other biologics. So these are just a couple of advantages that we have. And of course, we have been innovating in this space for as I said, 2 decades. So we own a lot of the IP around baculovirus manufacturing.
Great, thank you. And there's been some talk in the industry about growing pains as some companies, are upgrading their standards, so to speak, as they go from academia to testing in humans. Can you update us on your view specifically on the advantages of having your own manufacturing internally, which you've been working on for a number of years, and specifically you see advantages from your patented processes as well?
Absolutely. So we see baculovirus manufacturing and having the capacity to do this internally as being a key strategic advantage for us. And there are really two reasons. First, of course, is that for many gene therapy companies, they are sort of out of necessity born in academia. And so oftentimes the first clinical trial is run with a process that is developed, usually a hyperstack process that is developed at a university. This is often good enough to get kind of proof of concept studies, but is generally not reliable enough or inexpensive enough to actually be appropriate for larger scale studies and for commercial manufacturing. So for us, we kind of control the process from the beginning. And so we have not had to do these kinds of bridging studies that other companies sometimes have to do. Now, the reason I think that we really think that this is a key advantage is that not only, of course, does it make it faster, but in addition to this, we have been able to innovate over time. So we're slowly but surely driving the efficiency and the speed of the process and the cost of goods down to get to the point at which AAV manufacturing costs about as much as it costs to make an antibody. And so So I really do think that as we move towards larger indications like Huntington's disease and Parkinson's and some of the genetic forms of Alzheimer's disease, which are key areas for us, we will need to have the ability to make each dose much less expensively, and more reliably. And then as we move towards some of the indications where we have to move to the clinic quickly, the speed is also critically important. And so being able to, having essentially half of the company dedicated not only to making things, but to improving the process, I think has been, really a differentiating, strategic advantage for us. So I do think that this is a key part of the, of the story of uniQure.
Got it. And you mentioned preparing for the launch of etranacogene. So just update us, please, on the commercial manufacturing capabilities that you have in place. I think last year at our summit, there might have been a mention that your 200-liter scale process can commercialize etranacogene for a number of years. So where are we on what you have, what you need to do, and how you can commercialize successfully?
Absolutely. So our 200-liter process is absolutely ready to go. It is what we have used for all of our clinical trials. It is fully validated, and it is how we will launch etranacogene. And we have improved on that process. Of course, when you are about to launch something, you kind of have to freeze the process. So it'll be frozen for a few years, but we have now also have a 500-liter process that is also all, essentially ready to go. And at some point we will transfer the manufacturing from 200 to 500-liter. It's not going to be essential to do that until we have a, sufficiently large demand. For our subsequent programs, we are actually using the 500-liter process.
And so we've seen with actually, let me just start by saying there have been relatively few AAV-based gene therapies commercialized so far, but in the past, CMC has been a gating factor to get the product to market. So is that the case here? And could you let us know the general timeline for CMC-related matters as you move towards the BLA submission?
Yeah, so you're absolutely right. So in AAV manufacturing, we are sort of at the place where people were in sort of antibody manufacturing in the, in the 90s. There are still many parts of the process that are not understood, both in mammalian cells and in baculovirus. So everybody's trying to sort of get better process controls, better upstream and downstream processing. Having said that, of course, we have been running these studies for 5 or 6 years, so we have a fairly well validated process, and we're now in the process of doing the process validation for the BLA, as well as preparing for the inspections that are going to come. We're on track, and we expect to be ready to file as we have announced. I think that, but what you say is absolutely true, that for gene therapies, CMC issues are much more complicated than they are for small molecules and other biologics, mostly because it's just a much more complicated process, and there's just a lot of biology that has to work just right. And so I think, but having said that, I think that we are somewhat ahead of the curve when it comes to doing this.
Got it. And just a bit of a 101 on AAV gene therapy, please. What I'm getting at is, could you go into a little bit more detail about what you have to complete this year to submit the BLA? We just want to know how to track your progress. What are the things you have to do to satisfy the FDA, for example?
Yeah, sure. So in general, when you are going to do process validation, you need to have a certain number of batches have to fall in spec. You have to prove to the agency that you have control of your process, that you understand the process, and that you can make it for the market, and that you're not going to suddenly let the leave the market without a supply. And so to do that, you have to do these process validation runs. And so it's, you normally do 6 or 7 or 8 different batches. Of course, we've already done something like 40, so we need to do 8 more. And of those, we need, 3 have to come in spec in a row. Basically, that is kind of the requirement for AAV gene therapies. And then in addition to that, there's some stability requirements. So we have to do stability testing for the drug product. We have to do stability testing for, for the downstream material, and we have to show that it can be stored properly. And we're in the process of doing all of that. Of course, we've been doing it now for more than a year, but we're sort of at the final stages of that. And then there will be inspections, there will be GMP inspections, as well as there will be inspections of the GCP clinical standards. And that's kind of that. So that's sort of what's going to be happening. Of course, we're not going to announce exactly when any of these things happen. They are, dependent of when, on when the FDA, visits you. But they visit everybody, so I expect that they'll visit us.
Have an inbound question. Question is, how long does it take to do 3 batches in a row, etc.?
How long does it take to do 3 batches in a row? Okay, so
please teach us.
Absolutely. So each batch takes about 6 weeks. And, we have a certain number of lines that we can do at the same time. And so, it takes us, on the order of 4 months or something like that to do 6. Something, on that order. And we have, And then, of course, we have a whole set of assays that, including some potency assays. So we have a set of sort of release specs and so they all have to meet all the different criteria for release.
So again, one more 101 question, because this is relatively new, because uniQure is so advanced relative to the rest of the space. Now, stability, is that different from shelf life? What's the difference if they are?
Yeah, stability and shelf life are related, but not exactly the same. So you normally have to look at the stability of the sort of intermediates along the way, right? Because you will, for example, purify you have to purify what is called the sort of the drug product before you actually aliquot it into vials, and you have to store it. So you're looking at the stability of that. So it is sort of a shelf life, but really it's the shelf life during manufacturing. And then in addition to that, we have to look at the stability of the actual so there's the stability of the drug substance, and then there's the stability of the drug product. And the drug substance is an intermediate, and the drug product is the actual aliquoted drug. And so we have to measure the stability of all of that. And how stable it is depends on, as you would expect, on the formulation, and it depends on the temperature at which we store it. And so we have, actually worked on this for a long time, so we have some sense for how long we can keep it. And this is important because then, we will have to ship it to the different sites. But of course, we've run a trial, so we've dosed 60 or so people, so we have some sense for, what the stability is like.
Understood. And just roughly, how long might the shelf life be of an AAV-based gene therapy like China does? How long would you be happy with?
Well, I think so we store it at -80, and I think in principle the shelf life is really long. I think that the shelf life that we can claim really depends on how long we want to spend doing all the assays. I think we start with sort of 6 months of shelf life, and then we go from there. I mean, what most companies do, and what we will do, is we'll extend the shelf life as we, as we get more commercial experience.
Got it. And then I think you mentioned that the etranacog's margins could be as good as some biologics, in part due to the lack of plasmid requirement with insect baculovirus systems. Could you talk a little bit more about or quantify how costly plasmid is, just so we can get a sense of the advantage you might have?
Yeah, so in my previous life, I was at a HEC 293, a company that made AVs with HEC 293. So this was Novartis Avexis, and so it is generally the case that plasmids are the most expensive part, the most expensive sort of raw material. So they are, hundreds of thousands of dollars per dose. So while not wanting to, tell you exactly what the cost of goods are, because we've never made that public, baculovirus is significantly less expensive than that. And that I think gives us a real advantage.
Got it. And as I think you've also mentioned, insect cell baculovirus systems have been used elsewhere in biotech. But what I noticed is that 10,000 liter scale is also possible elsewhere in biotech using these systems. So is that the future of AAV, and how long down the line is that more driven by demand, for example, in the marketplace?
Yeah, well, I think that as we increase the scale, of course, the cost of goods goes down. And I think by the time you get to a 10,000 liter scale, we are going to be, the cost of each dose is going to be on the order of the cost of an antibody for each patient. So it's going to be, thousands of dollars as opposed to tens of thousands or hundreds of thousands as it is today. So to get to that, we have been working pretty hard on 500 and 2,000 liters. We, I think, need to perfect 2,000 liters before we can go to 10,000 liters. The good news is that people, of course, have been doing this for the vaccines and for other kinds of viruses. And so I think that we, at least the industry, has developed sort of expertise in, how to do this. And insect cells are really very forgiving relative to mammalian cells. So I am sort of cautiously optimistic that we can get there, but we also have to be careful to not overbuild capacity, because we want to somehow match our internal capacity to our portfolio. And so, so for now, we're building.
I think I'm not able to hear. Can you?
Oh, can you hear me? Am I muted?
Yeah, it could have been an issue with mine, but please continue. I think I lost the last 30 seconds.
Absolutely, yeah. So I was just simply saying that we want to make sure that we don't overbuild our manufacturing capacity. So I think 10,000 liters is entirely viable. It is something that we that is in our plans, and what I can tell you is that today we're working pretty hard on. So we have 500 liter ready to go, and we're working really quite hard on 2,000 liters. That already is a significant cost reduction. I think, but we're sort of being careful to not overbuild, because, we kind of have to build our manufacturing capacity at sort of the rate at which we have a pipeline. And so at the moment, in addition to Hem B, we have Huntington's, which is going to be significantly larger than hemophilia B, but still probably doesn't require much more than a 2,000 liter scale. So, and then, but some of the therapies after that will, in fact, require. I think 10,000 liters makes sense.
When you said Huntington's would be larger than hemophilia, are you talking about the market opportunity? Because that's not systemic. One would think it's less of a burden on manufacturing.
Well, I think the dose is smaller, but the number of patients is greater. So yeah, I mean, it's there are,
I'll punch it into my calculator later.
Yes, that's right.
So uniQure also has been talking about 200 liter wave bags and 500 liter stir tank reactor capabilities. So sounds good. So forgive me, I lied earlier when I said last 101 question, but please explain the concept, those concepts, what they mean for uniQure and the heme beef program and other programs.
Yeah. So the initial process that we developed used these sort of wave bag reactors. I think one of the things we learned was that and so our current process uses multiple wave bags that are effectively pooled before we do the downstream purification. We are moving to these stir reactors. They're more uniform, they're easier to scale, they're just more efficient. And that's, I think, the future for us. There are bigger wave reactors. I think what we found is that they don't scale very well. So we have sort of moved away from that. But also because if we really want to go to 2,000 and 10,000 liters, that really does require these giant stir tanks.
Got it. So is there danger that uniQure might have extra capacity? You sort of touched on balancing things, but to the extent you have the enviable problem of having excess capacity, maybe this is more of a question for Matt, but would you deploy that to partners or elsewhere? So first of all, how do you balance supply and demand? And second, if you have excess supply, what do you do with it?
Yeah, I mean, we have so far anyway really tried to balance our manufacturing with our pipeline as well as the programs that we have partnered with BMS and CSL. Our goal has really been to have our own pipeline and not to be a CDMO. However, if in fact we do build excess capacity, then we would certainly consider partnering with somebody else. The one issue, of course, is that for baculovirus, for the efficiencies of baculovirus to really work, you have to have developed your gene therapy in a baculovirus system relatively early. Otherwise, that transition could be a little bit painful. But yes, I mean, I think we would be willing to consider this. And I think we have now a lot of experience showing comparability between HEC manufactured AAV and baculo AAV.
Got it. And so for Huntington's disease, are you sort of following the same pattern as you did with etranacogene? For example, will there need to be a bridging study to port things over?
No, we don't expect that there's going to be a bridging study. I think for Huntington's, if we make any changes to the process, I think the product will be completely comparable. So I think one of the things that we've really tried to avoid is these bridging studies, which you often have to do, of course, if you're starting out with material that is produced, academically. But in our case, we almost always start with the final process from really the very beginning.
Understood. So there's a lot of flux or change ongoing in the industry. You could define that in many different ways, whether it's regulator, whether it's with processes, analytics. I mean, what do you think is changing and what are the most important advancements?
Yeah, I mean, from a manufacturing point of view, for sure, the biggest things are the learnings that we're getting from the kind of manufacturing of the COVID vaccines, which use some of the same technologies, certainly use the same supplies as I can tell you. That I think is, kind of accelerating, our capacity to do this at a much bigger scale. The future for AAV gene therapy manufacturing is, I think that kind of glide path looks a lot like the path that was taken by antibodies and other biologics. That is to say, we're still at the stage where, every batch is super expensive and super variable. I think we're learning a lot about the need to have in-process controls along the way, so we can kind of monitor both the amount and the quality of the AAV as it is being produced at the different stages. We're learning a lot about how to purify it. I mean, one of the key questions has always been, how many full capsids you have versus how many empty capsids, and how can we really purify full capsids more efficiently? And so, we're learning, as a field are learning to do this. We are developing antibodies that only recognize full capsids. We have, better epitopes. We have better ways of purifying things that capture more of the AAV. The productivity per mil is increasing, at least for us, and we have been pretty efficient at doing that. And in some cases, we've seen, 10x changes as we make really small changes in the process. So I think there is still plenty of room for that. So I think we're slowly driving out some of the noise so that the process is just more reproducible. I think it's not going to be quite the risk for every company. I think every gene therapy company today, as they get close to filing a BLA, starts to worry that the batches, the whatever 10 or 12 batches that they made during their clinical trials are suddenly not going to be makeable anymore because there are these things that they don't quite understand about the process. And I think that we're just getting much better at understanding exactly how the AAVs gets put together, and what fraction of the AAVs actually have the transgene. And in our case, for example, by not having to titer in. So we, for example, are moving away from having the 3 viruses that need to be infected and having just 2 and ultimately just 1, as well as having production cells so that the cells are actually stably infected, and this will make everything way more reproducible. So we're kind of driving the noise away. That's going to make everything more predictable. It's also going to make things more faster. And this also allows us to go to a much larger scale, which will reduce the cost of goods. I really do think that it's not going to be long before the actual cost of goods is going to approach the cost of goods of a normal biologic. That's really the future. And of course, that means that all kinds of indications that today are not really viable suddenly become viable.
So I'm intrigued by your comment that the COVID-19 vaccine rollout has actually helped some of the learnings with AAV. I'm just curious, though, whether in the short term, the demand for inputs there has driven up costs, at least short term, for what you're doing.
Yes. Yes. So in the short term, I think everybody has found that we're short of everything. We're short of bags, we're short of pipettes, we're short of, because it's, as it should be, there is a giant sucking sound to all of those disposables, and we all use the same things.
But then the companies make money selling them, and then you have plenty of supply. That's the way the world works.
Yes.
So then, just out of curiosity, just looking across the industry, because I know that uniQure is in some ways very different, but what do you see as the biggest CMC challenges in the gene therapy field right now? And then feel free to talk about how uniQure is positioned to address those industry challenges.
Well, industry-wide, for sure, the biggest challenge has been transitioning from the kind of scale that you need to do kind of first in human or small studies to much larger manufacturing. And many companies have tried to do this with sort of CDMOs. And I think it's a little bit of it's difficult to do that for the long term, because CDMOs are in the position of having to to serve many customers, and so it's a little difficult to actually optimize your manufacturing with the kind of to optimize that cycle time. So you have to make changes, and then and you make improvements, and that's difficult to do when it's a different organization. And so I think for us, it's a real advantage to be able to do this internally, right? And to have the people who are doing all the CMC development and who are ultimately doing the commercial manufacturing being right next door to all the scientists who are inventing the gene therapies. I think that has turned out to be a really powerful, a really a really powerful combination. So for us, that's a huge advantage. Of course, the challenge that everybody's facing, and there are quite a few companies now that are have been delayed because of CMC challenges, is that again, when you're in that process validation stage, you suddenly find yourself not being able to produce a product that is comparable to the product that you put into humans. Or alternatively, not having real control over your process. So, you develop some sort of a potency assay, and it turns out that potency assay is a biological assay and it has noise. And so there's noise associated with you making your gene therapy, and then there's noise associated with the assay, and then it becomes really difficult to make, to hit the release criteria. The same thing goes for other things like empty to full ratios. Again, unless you have really good downstream purification and you can really understand what it is that you're getting, you can get, weird mixtures of, full, partially full without, full with something that doesn't appear to be a transgene. So there's a lot of complexity because it is this complex biological process, and everybody's facing that. I would say the one thing I can say for us is that, we've been doing this since Globera. So we have made many, many mistakes. And so not that we don't continue to make some mistakes, but we make, fewer and, more exotic ones.
And what about the controversy of whether regulatory standards are getting tougher, which is one perspective. Another is that as, there's now dozens of companies, public and private. And so if the regulator sees a higher standard, it wants the industry to adopt it, which is in a way tougher standards as well, versus there were publicly traded academic vehicles, especially years ago, less so today, and they made mistakes. And so that's going to die down as the industry's professionalized. Which of those themes resonate or offer any alternative themes as well too?
I think they're both good insights. I think as the field has matured, the agency has just seen many more packages. So it has much more experience seeing CMC, and so they kind of know what to ask and what to look for, and they can. And to some extent, AAVs are kind of a de-risked drug class, right? So if you have a small molecule, every small molecule is its own horrible adventure. But if you have a gene therapy, you kind of understand what is common of all gene therapies, and they all have sort of the same kinds of things, right? So for all of them, you have some sort of a potency assay, and for all of them, you need to understand, the kind of particle size and which ones are full and which ones are empty, and that kind of characterization has spread throughout the field. And so when you show up now with, something that looked pretty good in a sort of academic, non-human primate study, and you put it into 3 patients, they're not as enthusiastic about allowing you to do a much bigger study unless you can kind of bring it up to the standards of the whole field. But the other thing that is really that is creating a sort of shared set of standards is that there are now quite a lot of CDMOs that will do the manufacturing. And of course, they all have access to multiple different companies, and they're all creating this sort of uniform playing field, which I think is just professionalizing the industry. I mean, I hope that we will get to a point in AAV gene therapies where the thing that you're really worried about is whether your drug is going to work, not whether once it works, you'll be able to make it, right? Which is where many companies are.
Understood. So on the topic of, let's say, systemic tox, and I know that AAV5 has been very fortunate in terms of the signals. It's pretty good so far. But keeping in mind that that's a sector issue, and you're operating from a luxury position, let's call it, what can manufacturing do to reduce the risk even further? Was that clear?
Yeah, sort of. I don't know that this is really a manufacturing issue. I mean, there's definitely different kinds of toxicity associated with different AAV serotypes, right? So, fortunately, we've been relatively fortunate with AAV5. I mean, I think the trade-off for AAV5 is that it's somewhat less infectious, so it's a little less potent, but it's also less immunogenic. So, you tend to not generate quite as many, or in fact, any T cell responses, and not too many antibodies. And that means that, we can dose it without having to pre-treat people with high-dose steroids. And that is also true in the CNS, whereas for, AAV8, people make really pretty strong responses, and there are some fraction of patients that, the safety margin gets a little narrow. And, for AAV9 and AAVrh10, there may, in fact, not be a completely zero effect level on NOEL. But I'm not sure that that's really a manufacturing issue. I think at least most of that is a serotype issue. The one thing that is a manufacturing issue that continues to be hugely controversial is the role of empty capsids versus full capsids. And that, I have to tell you, I have been studying this for about a decade, and I still don't know whether empty capsids matter or not. And I can point to, studies that seem to show that, if you have a lot of empty capsids, it might increase the number of antibodies, but then I have just as many studies that suggest that it makes no difference. So it's a little complicated.
So just in your manufacturing forums, internally and externally, you're not hearing things like post-translational modifications or impurities, things like that are contributing to systemic tox. It really does sound like it's mostly capsid.
Yeah, I think, so unlike antibodies, right, where like glycosylation is really important and things like that, let me just say this. I think one of the problems is we don't have that level of characterization. So we don't really know what the post-translational modifications are on like the various capsid proteins. So therefore, it's a little hard to know whether any of them are associated with toxicity. I think most people don't have a completely homogeneous product. And as a consequence, we don't know what the effect is. So we don't really know, for example, why it is that, some companies saw, talks with, AAV8 in the liver, and for some reason with AAV5, you don't see toxicity. We assume that it's because of the serotype, but of course we make ours in baculo and they make theirs in HEC. So I guess theoretically it could be that. I think to really know for sure, you'd really have to do side-by-side comparisons.
Got it. And then just curious about the future and what important innovations in manufacturing that you're looking for that'll better enable you to meet or exceed your goals.
Yeah, so I touched on a few of those. From a manufacturing point of view, I think, a big part of that. So, you're always trying to improve three things, right? You're trying to improve reliability, you're trying to increase the speed, and you're trying to decrease the cost, right? So what can you do to do all of those three things? I think in terms of reliability, there is a lot you can do in terms of sort of downstream purification, because I think most people find that being able to control the kind of upstream conditions, is pretty difficult. Whereas being able to isolate the full capsids is a sort of an easier proposition. So we're getting better at that, and that I think is pretty important. And moving beyond the kind of relatively simple-minded columns that we have today to slightly more sophisticated columns and more sophisticated purification protocols, or moving away from cesium gradients, which is what some people use. That is, I think, something that we're doing. Then, and so that should improve the reliability. When it comes to improving the speed, I think for us, it's really, as we learn, because the only thing that you really change from one AAV therapy to the next is the cargo plasmid or the cargo virus, right? In our case, the baculovirus. And so really trying to understand how it is that you put in a cargo virus and it essentially generates the same product, right? If it's just a question of substituting this one thing, then we don't have to re-optimize it every time. And that's just going to make the whole CMC process so much faster. And then from a scale point of view, of course, for baculo, we're already in suspension. So we already have a fantastic suspension line that grows super densely. We, have these SF plus lines, and we're always making better ones. But basically, but we're now getting to the point where instead of having to use 3 baculos, we can use 2 or 1, or we can use just 1 in the context of a production cell line. And I think that is going to be a big deal, because it's going to make everything just much more reproducible. And in hex cells, of course, people are also trying to make cell lines and stable cell lines. Again, it's been really, really hard to make a complete stable cell line, but you can make a cell line that has, maybe the rep and cap gene stably integrated, and you only have to put in the transgene. And again, moving away from adherent to some sort of a solution-based system is also a huge advantage, because you can grow those cells at much higher densities. So yeah, so those are some of the innovations that everybody's working on and that we're working on as well.
Great. Thank you, Ricardo, for all the great insights.
Sure. Thanks so much for the invitation.
You're welcome. We now conclude this session.
Bye.